So it really took until the late 1990s when we were doing things that understood what really drove cancer, specific mutations in specific kinds of cancers, led initially by a colleague of mine who was here, Brian Drucker, who had this idea that mutations in a type of leukemia could be a good target to shut down. And the rest is, of course, history, that this drug called STI-571, then Gleevec, shut down that signal in these leukemia cells and basically made chronic myeloid leukemia a disease people could live for decades with rather than die from.
With the advent of recombinant DNA and DNA sequencing technology, we have gone from things like X-rays, toxic chemicals that do indeed kill cancer cells, but they kill any dividing cell. We've gone to the era of precision medicine. We can develop medicines and some antibodies that are specifically configured to kill only cancer cells.
EGFR stands for epidermal growth factor receptor. It is a receptor that is found on the surface of many lung cancer cells. In the early 2000s, we were able to discover that a subset of individuals' cancers have a mutated form of this receptor. And this mutated form of the receptor keeps the receptor on continuously, kind of like your pedal and the gas continuously in the car, and as such, the spurs on the growth of cancer cells. In some individuals who have these alterations, therapies that were meant to stop the receptor were particularly effective in patients whose cancers have these genetic alterations.
And the other huge field today is the immunotherapy, the ability to harness the immune system, which is meant to protect us from diseases, including cancer, and to turn it around and to use the immune system to fight back against cancers.
I've been involved in lots of different types of therapeutics, whether it's immunotherapy, antibody-drug conjugates, novel oral agents like CDK4/6 inhibitors. And a lot of my research interest is to figure out how to tailor therapy so that we give the patient the right amount of treatment for their cancer. We're not over-treating them and giving them too many side effects, but we're giving just the right amount that's going to lead to the best outcomes for that patient, but also decrease the amount of toxicity that we're giving. And so that has driven a lot of my research, is this idea of sort of right-sizing therapy to the individual patient.
What successful immunotherapy is stopping the cancer from turning off the immune system. And it's now one of the pillars of cancer treatment. And that's basically because we've learned to block the PD-L1, PD-1 pathway. And this is now FDA approved in 21 different types of cancers, and probably with more to come.
Well, when you think of the future of immunotherapy, you have to remember that still only 25% of people respond to current immunotherapy. 25% is not enough. We want to be able to treat 100% of our cancer patients with immunotherapy. And this is why it is so important that Dana-Farber has such rich, talented researchers. We do so many clinical trials here, 1,100 a year. And that's critical not only for research, but for our patients, because 20% of our cancer patients have no options except to be on a clinical trial.
We really want to be able to have effective therapies for all patients that have cancer, and more importantly, be able to detect cancer early enough that it can be easily taken care of. We're on a path to get there. I don't know how long it'll take us to get there, but ultimately I think that's the goal.